Have you ever looked at a naked mole rat and asked yourself, wow, I wonder how this truly beautiful creature is able to live completely cancer-free for over 30 years? Well, you've sure come to the right place to find out. Welcome everyone to The Protein Show. Today's episode, brought to you in part by our good friends at Cambridge Analytica, is all about human hyaluronidase 1. Hyaluronidase 1 is a member of the hyaluronidase family, and a member of the greater glycosyl hydrolase superfamily, which, as their name might entail, catalyze the hydrolysis of glycosidic bonds and sugars. Hyaluronidases are a class of proteins that predominantly degrade hyaluronan. Hyaluronan, which is also known as hyaluronic acid, is the incredible molecule responsible for the naked mole rat's cancer resistance mechanism and amazing lifespan. Hyaluronan is an anionic or negatively charged glycosaminoglycan, which are a set of long, charged polysaccharides made up of repeating disaccharide units. Unlike heparin and many other glycosaminoglycans, hyaluronan does not have a sulfate group. As mentioned earlier, hyaluronan is composed of repeating disaccharides, which are themselves composed of D-glucuronic acid and N-acetyl-D-glucosamine. Hyaluronan is a major component of cartilage and is extremely important in joint lubrication. Hyaluronan is also found in a variety of other tissues in the human body, and is also heavily implicated in cell proliferation, stem cell differentiation, cell-cell recognition, and most interestingly, tumor growth. However, what might be most unique about this sugar is that it can have an incredibly variable molecular weight, ranging from 5 kilodaltons to over 20 million daltons. This range of molecular weight is in part what researchers in Rochester's own Gorbunova lab have been studying for close to a decade. In the naked mole rat, for example, hyaluronan has an average molecular weight between 6 and 12 million daltons, which is thought to contribute to their remarkable cancer resistance mechanisms. These incredibly long, viscous hyaluronan molecules are maintained in the naked mole rat through upregulated anabolic mechanisms and downregulated catabolic mechanisms, which is where hyaluronidase comes in. Hyaluronidase 1, the most prominent of the 5 hyaluronidases encoded in the human genome, was a 435 amino acid residue protein discovered in 1928 by Dr. Francisco Duran-Reynolds. These specialized enzymes hydrolyze the beta-1,4-glycosidic bond of hyaluronan into various oligosaccharide lengths, the shortest being tetrasaccharides. Hyaluronidase 1 is primarily responsible for the breakdown of intracellular hyaluronan. This enzyme is found in the lysosome, the organelle charged with the breakdown of many biological macromolecules. Its location in the lysosome is consistent with experimental evidence of acidic pH activity, somewhere in the range of 3 to 4.5 pH units. As you can see in this animation, hyaluronidase 1 is composed of both beta sheets and alpha helices, forming a distorted alpha-beta barrel composed of 8 of each secondary structure. The enzyme contains 2 domains, an N-terminal catalytic domain and a smaller C-terminal domain. Hyaluronidase 1 also contains an interesting EGF-like domain, which is an evolutionarily conserved enzyme domain present in a wide variety of proteins in the human body. While the exact function of this domain in hyaluronidase 1 is unknown, it is postulated that it interacts with the CD44 antigen, a glycoprotein involved in cell-to-cell recognition and interaction, which acts as a hyaluronan receptor for the cell. The enzyme is also very heavily glycosylated. Indeed, its structure shows multiple N-linked interactions with alpha and beta D-mannose, as well as glycerol, and N-acetyl-D-glucosamine, one of the sugars that makes up hyaluronan in the first place. Stabilized by hydrogen bonding and hydrophobic interactions with nearby amino acids, it is thought these oligosaccharide modifications may assist with folding stability and potentially the functionality of the enzyme itself. In terms of its mechanism, it seems that hyaluronidase 1 is able to hydrolyze glycosidic bonds through a nucleophilic attack by an acetyl oxygen on carbon 2. This is thought to be followed by a proton transfer from a glutamic acid residue to the oxygen on a neighboring carbon 4. A water molecule then enters the active site, and through polarization by a glutamic acid and tyrosine residue, it is able to complete the hydrolysis to nucleophilic attack at carbon 1. This occurs in the enzyme's active site, in a small cleft in the N-terminal catalytic domain. The position and size of this cleft is thought to produce the enzyme's tetrameric product specificity, which is seen in most other mammalian hyaluronidases. Interestingly, multiple splice variants of hyaluronidase exist in the human body. As shown in this figure, splice variants 1 through 5 lack certain aspects of the full hyaluronidase 1 enzyme. While the full enzyme is present in most tissues in the body, researchers have found elevated levels of certain splice variants in very early prostate and bladder cancer cells. Later stage cancers, however, have evidence of increasingly abundant full-length hyaluronidase 1, leading to higher levels of short-chain hyaluronan, which is thought to ultimately contribute to cancer formation and proliferation. While high molecular weight hyaluronan has been found to grant the naked mole rat cancer resistance and an incredibly long lifespan, the need for additional research on this molecule and its catabolic enzyme, hyaluronidase, has never been greater. Increased understanding of splice variants and the regulatory transcriptional mechanisms of hyaluronidase may allow us to further understand the spread and distribution of some cancers. Hyaluronidase's incredible importance in cancer regulation ought to be further studied. Indeed, researchers have suggested that increasing the abundance of certain splice variants may be a viable strategy to consider when researching novel cancer therapies, potentially helping to save millions of lives, all with a little help from our good friend, the naked mole rat.